Imagine piecing together humanity's family tree, tracing migrations across continents, or finding the ancient origins of diseases that still plague us. Forget dusty old books. To truly understand our deep past, we turn to ancient DNA. This is where science resurrects genetic echoes, transforming dust and bone into the vibrant story of our ancient journey. Welcome back, time travelers. We've seen DNA collected from crime scenes and ancient digs, then amplified with shared lab tricks. Now let's jump specifically into paleogenetics, uncovering the unique techniques and mind-blowing discoveries that let us decode our ancient ancestors' very genomes. Working with ancient DNA isn't your average lab day. It's more like molecular archaeology. Every fragment is a tiny, precious clue, often bearing the deep scars of time. Biggest headache? DNA degradation and fragmentation. Thousands of years turn long DNA strands into tiny broken pieces. Plus, there's postmortem damage, especially deamination, where DNA's C's turn into T's, tricking our readings if we're not careful. Another huge pain: low endogenous DNA. Imagine a bone fragment filled mostly with dirt and bacteria, with maybe less than 1% actual ancient human DNA. And of course, the constant fear of modern human contamination. One tiny skin cell from a scientist can ruin everything. That's why those super clean labs from Part 2 are non-negotiable. So, how do we hack it? 1. DNA repair enzymes. We've got special enzymes to fix some of that ancient damage before we read the DNA. 2. Optimized library prep. Our lab kits are super efficient at grabbing every single tiny DNA fragment. Number 3. Targeted capture. We often use molecular baits to literally fish out the ancient human, or whatever we're looking for, DNA from the massive soup of microbial junk. It's like finding a needle in a haystack, but we have a super magnet. Once that ancient DNA is prepped, it's decoding time with next-generation sequencing, NGS. This tech reads millions, even billions of tiny DNA pieces at once, perfect for our fragmented ancient samples. It goes like this. We sequence the fragments, then use heavy-duty bioinformatics, that's fancy for computer analysis, to map those tiny damaged reads to a reference genome. A crucial unique step for ancient DNA is authentication. We literally look for those telltale damage patterns like the C to T changes and super short fragment lengths to prove the DNA is genuinely ancient, not modern contamination. If it doesn't have the battle scars of time, it's a fake. Once authenticated, we start piecing together the ancient genome, even if it's just a partial snapshot. Armed with these tools, paleogenetics has exploded with discoveries, completely rewriting history books. 1. Human migration and population history. Ancient DNA has mapped human migrations across continents, showing how waves of farmers and herders shaped Europe's genetics, or tracing the complex journey of early Americans back to specific Siberian populations, like with the Anzac I child. We can literally track our ancestors' movements across millennia. 2. Neanderthal and Denisovan admixture. Mind-blowing revelation. Our species, Homo sapiens, didn't just replace Neanderthals and Denisovans, we interbred with them. Ancient DNA proved many non-Africans carry 1 to 4% Neanderthal DNA. This flipped our understanding of human evolution from a straight line to a messy, fascinating family tree. 3. Evolution of ancient diseases. We can even study ancient pathogens directly from remains. Sequencing Yersinia pestis from Black Death victims, for instance, showed how the plague evolved through history. This gives us crucial insights into diseases like tuberculosis, smallpox, and even the origins of hepatitis B. Number 4. Ancient traits. We can infer what ancient people looked like. DNA has revealed the eye, hair, and skin color of early Europeans, showing some had darker skin than we imagined. It even tells us when traits like lactose persistence, drinking milk as an adult, evolved, often linked to new farming practices. Paleogenetics is constantly innovating, squeezing more secrets from the past. Future frontiers include more complete ancient genomes. We're getting closer to routinely assembling high-quality whole genomes from more ancient individuals, even from challenging environments. Environmental DNA, eDNA, for ancient ecosystems. We can now analyze ancient DNA from just soil or sediment, rebuilding entire ancient ecosystems, the plants, animals, and microbes that once thrived there without needing to find a single bone. Non-destructive sampling. Scientists are developing ways to get DNA without damaging precious artifacts, preserving them for future generations. Metagenomics. This is like analyzing all the DNA in a sample, human, microbial, and environmental, to get a complete picture of ancient diets, health, and ecosystems. As paleogenetics advances, ethics remain critical. Respect for human remains, collaborating with indigenous and descendant communities, and ensuring responsible data handling are vital to ensure this incredible science benefits everyone. From these microscopic clues, paleogenetics offers an unparalleled window into the profound story of life on Earth. And speaking of cutting-edge DNA analysis that impacts living populations, our next episode will jump back to the present with a powerful forensic application: forensic genetic genealogy, where ancestry meets criminal investigation to solve the toughest cold cases. Make sure to subscribe to the Geneticist, like this video, and ring that bell to catch all our deep dives into genetics. See you soon.